For the introduction. Welcome, everybody. So my talk today is kind of twofold. First of all, I want to talk a little bit about scalable web-based multi-volume rendering data structure that we have developed like 1-2 years ago. And then on top of such multi-volume rendering, I want to introduce a cell-to-cell interaction analysis tool for such data. So here we see a 2D whole slide immunofluorescence imaging dataset, and 2D whole slide immunofluorescence imaging allows us to improve our spatial understanding of biological tissue microenvironments, right? But it has some limitations due to the relatively low single cell resolutions of a few pixels. And also we miss some important information regarding the shape and the surrounding of the neighborhood of cells because we're looking here at a 2D slice. So in this work here, the scientists basically for certain sections image that in 3D with higher resolution, and that of course allows them then to look into individual cells, their shapes, and also like in the interaction of these cells in more detail. So basically over the years, I had been working with two of these SciSafe 3D datasets. One is smaller. This is around like 1,000 times 1,000 times 40 pixels and 2.4 gigabyte in size. And then another one that came later that quadrupled basically in each of the three dimensions and also has more channels. And it's a factor of 60 in terms of the gigabyte size here. So when you think about rendering such imaging data, then of course, there's some requirements. First of all, you don't want to have that data locally stored on your hard drive or so. So we kind of want a client-server architecture where the data is on the server and then loaded to the client on demand. And then the approach should be scalable, right? Scalable in two ways. First of all, we would like to have an out-of-core rendering where we do not have all the data in memory, basically, but just load on demand what we currently render from given our viewport. And then also we want to have multi-channel support, right? Because the high computational cost of raycasting grows with the number of channels. So we want to be scalable in that sense. Okay, so the first approach I want to talk about is residency octree, a hybrid approach for scalable web-based multi-volume rendering. And I want to especially emphasize that this approach or this data structure and algorithm was developed by Lukas Herzberger. He implemented the work and it was supervised by the people here on the slide, especially Markus Hartwiger and Johanna Bayer. And me, and of course also the PIs from the different labs collaborating here with each other. So when a volume is too large to fit into memory, there are basically two obvious strategies, right, that we can come up with. We can divide it into bricks, and we can downsample it to get a multi-resolution hierarchy. And with this, all that's really left to do is to figure out which bricks are actually needed for the rendering. So for this purpose, state-of-the-art approaches employ kind of ray-guided methods, which make use of the fact that the rays don't travel through the whole volume, as you can see here. Instead, some bricks might not be visible because of the current viewpoint or occluding structures in the volume, right? And ray-guided methods record those bricks that are visible during rendering and stream them to the GPU where they are stored in a brick cache. Now, currently, ray-guided methods are either based on octrees or they're based on page tables. So in an octree-based approach, there is kind of like a one-to-one mapping between bricks in the dataset and the octree node. And so each node corresponds to exactly one brick and vice versa. To access a brick during rendering, now we can start a traversal from the root node. And at each step, we check if the node's brick is currently in cache, right? We use the last node that has the brick currently stored in cache for rendering. In case a node's brick is currently not in cache, we can simply use a previous node's brick instead. So the good thing here about octrees is that we can substitute the high resolution data using a lower resolution as long as we don't have it loaded, for example. The downsides are that we have to store lower resolution in cache, even if we only ever render the high resolution data, and that we have to do this traversal every time instead of accessing cache bricks directly. Page tables, on the other hand, draw their inspiration from memory virtualization, and they simply store for each brick if it's currently in cache. And this allows them to provide direct access to any of the brick that is currently in cache without the need of a hierarchy traversal. Now, the downside of this is, of course, that we don't have a clear strategy here for substituting bricks that are currently not in cache. Like octrees, page tables can also be used for empty space skipping, but only supporting skipping empty bricks, and are thus kind of like a little bit limited when it's compared to octrees. So wouldn't it be cool to have the direct access that page table provide, but also have the security of octrees when it comes to skipping empty space and substituting missing data? And that is where our resident octree comes in. So the problem again with octrees is that bricks and nodes are intrinsically coupled. So we have this one-to-one mapping between them. And now with our approach, the residency octree, we decouple the spatial or geometric subdivision determined by the octree from the resolution level in the volume dataset. So instead of mapping each octree node to a single brick, we map each node to a set of bricks in each resolution level. And that allows us to use the residency octree for empty space skipping for a per node basis instead of a per brick basis. And it also allows us to evict unused low resolution data from the brick cache if we only ever render high resolution data and vice versa. So now let's just get a feeling of how these three methods behave in terms of their cache utilization. So this is like a data set about a king snake here in high resolution, and we visualize the number of bricks that are strictly required to be in cache per pixel by each of the three methods. So brighter pixels mean more bricks. So when we use the standard octree approach, and we render this, we can see that around the snake skeleton, we have a lot of bricks that are required to be in cache. And that's of course because low resolution bricks need to be in cache so that the higher resolution data can be in cache too. However, outside of this region of interest, we see that the volume is nearly empty here. So almost no bricks have to be cached because the entire octree nodes can be skipped already in a higher level. Now with the page table, high resolution bricks are accessed without a hierarchy traversal, and low resolution data is not required to be in cache, right, as you see here. However, because of their limited empty space skipping capabilities, more non-empty bricks outside the region of interest are kept in the memory. Now, finally, with the octree approach that we developed, we can converge to a state where we only have to keep those bricks in cache that actually contribute to the output image. And so the translucent bricks and low resolution bricks that are never used based on the current rendering settings do not have to be kept in the memory. Okay, so now let's just quickly look how that extends to multi-volumetric data, because we do not only have one volume. And then we can look into some of other data sets and like some performance measures. So in the single channel case, the residency octree stores for each node in which resolutions its corresponding bricks are currently available in cache, right? We discussed this. Now, when we extend this concept to multi-channel rendering, this is kind of trivial. So instead of storing the cache residency of corresponding bricks for just one channel, each node now stores it for multiple channels. So little illustration here. So we can start the residency octree traversal at the root node and traverse the tree until we reach an empty node or a leaf node. And then we can process each channel individually. So we might, for example, for the blue channel, access the medium resolution, for the red channel, the highest resolution, and maybe we choose for the green channel, maybe it's less important, we select low resolution. And so in all of our benchmarks that we have here, not all of them are like immunofluorescence data sets. Some of them are just like data sets that we find out there as like a benchmark. And we can also basically create artificially multiple volumes out of that. But we also have two decisive data sets here in our benchmarks, right? And in all of them, the residency octree outperforms the page table and octree, both in rendering performance and GPU memory usage. And that is because it supports more fine-grained empty space skipping, which is independent of the bricking granularity in the dataset. And it's also independent of the resolution level in the dataset. So basically, we can represent any node in any resolution and access any brick from any resolution directly. And that's why we do not require more bricks to be in memory than necessary. I also want to point out the GitHub repository here with the Rust implementation of this. Now, this is not in a state, I would say, to be integrated directly into an application, because there are still, like, it was enough for this research paper, but there's still like some open challenges in terms of like capability with the integration in different browsers, security things, also some aspects on the algorithm and so on. But I heard that some or part of it might be considered for VUES, and so that's nice to hear that that will hopefully be adjusted, adapted, and made more useful for the community. So I want to highlight here the SciSafe small data set, where you can see basically in terms of GPU memory usage, which the residency octree brings down to kind of like a third compared to page tables. And now on top of such multi-volume rendering approaches, one can build visual analysis capabilities. In this case here, we focus on interaction analysis. So we wanted to look like how cells interact. This is also a collaboration between different labs here at Harvard. You see many familiar faces here, and the University of Vienna. And I want to also highlight here Kevin Zidak and Eric Mert, who implemented like hands-on on this tool. So thank you so much for this. And they basically realized this work. Okay, so what do we focus on here? Well, the approach focuses on understanding cell-cell interactions, right, in the cancer microenvironment here, specifically the interaction between immune cell and cancer cells, because the hope is that by understanding this better, we can identify targets for novel immunotherapies at some point. And here on the left side, this is an illustration of a 2D data set where interaction analysis is often done based on proximity. So you look like which other cells are close to a cell, and then basically you infer interaction because cells are close to another, right? And every cell has only a few pixels. So there is maybe a little bit more than proximity to see here, but not enough to really look closely what's going on between those cells. Now in the 3D space, we have a much higher resolution and also like another axis, another spatial axis. And so we can basically trace the cells from one cell to the other. We can trace the voxels and look really what the protein distribution is between those cells. And this is a work inspired by the paper from Nirmal et al., where they basically did this as part of their overall analysis of these data sets. And we basically wanted to make this approach a little bit more, I would say, scalable and integrated into a visualization tool. So here is our volume viewer. And this volume viewer here, I have to admit, does not yet use the residency octree because it is not ready to be integrated in this tool yet, but it loads a smaller volume, so it's fine here. And you can see that in this case, we have also, we can visualize multiple volumes, in this case here DNA, then CD163, immune marker, SOX10 cancer marker, and then basically the viewer allows you to activate and deactivate different channels, zoom in, pan, look at from different angles, and apply certain transfer function for the pseudo color mapping. In this case here, we kind of also see here this PD1, PDL1 pathway between T cells and cancer cells. Okay, so you already spotted that there's a graph here, right, between these cells. That's basically a scaffolding that defines the interaction space. So we did not have segmentation at the time for the data, so we chose to go with a segmentation-free approach, where the user basically just needs to label the cell centers. In this case, this was done in Elastic. And then we construct this Gabriel graph here, which kind of connects nearby cells with each other. And now we can start tracing the volume. So basically, literally, the algorithm goes from cell 1, voxel by voxel, along this edge to cell 2. But it does not only consider the voxel directly on the edge, but with a certain radius around it. So basically, this is this cylinder here on the right that we span depending on the radius that we consider between the cells. And then basically we integrate all the voxels onto the edge and we get these line profiles. And as you can see, we can also select different shapes like a bicone structure that puts more emphasis on the space directly where the cells meet, where the cells meet. So the user can choose what kind of shape to use. So here we can see that in action. So in this case on top, we have this cylinder and we basically trace the this volume in there from B to A, and that gives us that line profile, and the line profile looks slightly different for this bicone shape here. But we can do that for every edge, and then we basically end up with profiles for every edge, for every interaction that we can then, of course, also sort based on their similarity. Another phenomena of interest for our collaborators was polarization of cells. So basically a more cell-centric view. Instead of looking what's going on between cells, we basically look how are the proteins distributed within a cell. And again here, because we do not have a segmentation in this case, here we made the assumption of a sphere and basically look at all the voxels and the intensity expression within that sphere and project it down to this viewing plane that you can see here. So this viewing plane is dependent on the current camera position so that the user sees, it aligns basically what the user currently sees. How do we encode this? Well, basically we have these different orbits. For every channel we have an orbit, and we can express how strongly a protein is expressed in which angle from the center of the cell. So this is how it looks then in the tool. And you can also, of course, interact with it in 3D space. And when you stop the interaction, then the viewing plane will be updated based on the current viewport. Okay, so we have a kind of like a pipeline here. We have the multi-volume channel data. First of all, we construct a graph, and the user can then explore this and also make slight adjustments to the graph. And then we have two things. First of all, the interaction analysis, where we focus on what's going on in terms of the protein expressions between cells. And then we can have the polarization analysis. Now, for the interaction analysis, when we look at the few channels only, we can express that with like line profiles. But when we look at many channels, that gets a little cluttered. So we choose a basically more clutter-free, more compact representation as a heat map. For the polarization, we can show that directly in the image space. But again, like when we have many channels, we have a lot of these orbits, and that overlaps a lot of the volume. So we can then also present that as a heat map where we basically unroll this kind of radial chart into a heat map. So this is how the tool looks like. In the center, again, we have our volume viewer, which is now surrounded with these interaction profiles. And here on the right side, we have a rendering panel, basically, where we can choose which channels we want to have active. We can set the transfer function, pseudo-coloring, and so on. So we performed a couple of case studies. Based on the time, I just want to go into detail for one of those. So this study here focuses on quantifying immune suppression of T cells by tumor cells via the PD-1, PD-L1 pathway. And in this example, we basically see this cell B here. This is like a T cell. And we see cell A, a cancer cell. And then when we look at the edge E1, we basically see this PD-1, PD-L pathway here of the immune suppression. And we also see this on the edge E2, but it's not present on edge E3 because these are basically two cancer cells here. So we can then quantify this with these line profiles and then compare them and sort them based on similarity and so on. And you can also see here in the center that this polarization basically gives a more cell-centric view in terms of like in which directions markers are exposed. We had a second case study here on macrophages, but I think I will skip that for now. It's all in the paper basically, but you can see the tool in action here, and this one involved many more cells, so it demonstrates also a little bit the scalability of the tool. Okay, so as a conclusion, 3D high-resolution imaging enables novel analysis possibilities, and I presented a data structure that we call residency octree to efficiently render such multi-volumetric data at scale. On top of such rendering can sit different analysis tools. For example, the one that I showed, cell-to-cell, which focuses on the interaction and polarization of proteins. And for the future, I think we should probably step away from this kind of cell counting, voxel counting, and use more like segmentation-based approaches with deep learning, and for hopefully extract more rich features with these deep learning approaches. And I think visualization can enable exploration and also explanation of the features that we detect here. So I want to say a special thank you again to Lukas Herzberger, Kevin Sidak, and Eric Mert, because they implemented on these tools. And I also want to especially thank Clarence Yap for the imaging. This is like exceptional work, and I think without the imaging, none of this downstream analysis work would have been possible. Also, Julia Valius for being available and for case studies with the data, and Sandro and Ajit for their advice. And of course, all the co-authors and the PIs in the labs that we work together with. Thank you so much.
Thank you for this talk, Robert. Are there questions?